Alrighty everyone, welcome back for our next lab presentation. In this presentation we're going to cover the special senses. Now the special senses build upon what we learned when we discussed the general senses. And as human beings we have a few very specific special senses that we're going to discuss throughout this unit. The special senses topic will be separated into more than one video, and in this video in particular we're going to cover the eye and vision. Vision is in fact a special sense, and vision is facilitated by the eyeballs. But a few other examples of special senses that we're going to discuss throughout the course of this unit are olfaction, which is our sense of smell, and gustation, which is our sense of taste, as well as our sense of audition, which is hearing, and balance and equilibrium. In total, we're going to learn 5 different special senses, but we're going to get started with the eye and what its roles are. So to get started on this special sense, we have our organizational slide laid out to discuss a few very important parts of the eye. In particular, the eye is separated into different coats or layers. We have an outer layer, a middle layer, and an inner layer. But each layer is composed up of different things. So the outer layer is made up primarily of what's known as the sclera. The sclera is the white part of the eye, and I will elaborate on this structure's functions very shortly. We also have the uvea, which is the heavily pigmented layer of the eye, which is also going to contain the ciliary body, the choroid, as well as the iris. And we'll discuss the functions of each of these parts as well. And then the inner layer is the retina. The retina is a very specialized layer that contains photoreceptors. And photoreceptors, as they sound, are receptors that take in light and send those signals back to the brain so that we can make a picture out of the light that has entered into the eyeball. The photoreceptors that the retina contains are called rods and cones, and I'll briefly discuss those as we make our way through this presentation. So this slide really should just serve you as organization. We're going to go over each of the anatomical parts of the eye and their functions, and then we're going to move into some visual testing methods as well as some pathologies. So first and foremost, we're seeing a sagittal section of the eyeball. We have this outermost layer here, which is the sclera, and the sclera, I didn't mention on the last slide, does have one specialization known as the cornea, which we'll see over here. We have the uvea, which is found here. This is shown in red to indicate that it's heavily pigmented, but this part of the uvea is the choroid, which is going to be highly vascular. We have the ciliary body shown right here, and we have the iris. All of these are within this same middle layer, but there's different parts to that layer. So we're going to go ahead and talk about each of those. And then the inner layer, which is the retina, is this yellowish layer that can be found here, lining the inside of the posterior chamber. So let's break down each individual part of the eye and see what each one of these structures does. So first and foremost is the sclera. The sclera, as I already mentioned, is in fact the white part of the eye. So you see this every time you look into the mirror. But the purpose of the sclera is not to contrast with the colored portion of the eye to make us look good. The purpose of the sclera is to provide structural support and to provide shape to the eye. The sclera is a very hard layer, and it's also going to protect the structures that can be found towards the inside of the eye. The eyes are in fact very fragile organs, and the sclera help to mediate their interaction with the external environment, and the sclera help to mediate their interactions with the external environment. Now the sclera is going to also be continuous with one of its specializations known as the cornea. So the cornea can be found right here. The cornea is in fact a transparent part of this layer, so the sclera will not be transparent and you won't be able to see through it. The cornea will be transparent and it's going to allow light to enter into the eyeball. This will unlikely pertain to all of you, but those of you that wear contact lenses will actually be placing your contact on top of the cornea. I myself am a very avid contact lens wearer, so I'm very accustomed to touching my cornea with the contact lens. So the sclera provides structural support, and the cornea is going to be transparent and helps to refract light into the eye. And I am going to be using that word frequently throughout this presentation. Refraction for our purposes just means reflection. So this cornea is just going to reflect light onto this big structure, which is eventually going to focus it back here onto the retina. Now our next deepest layer is in fact the uvea, but this part of the uvea is known as the choroid. The choroid is in fact heavily pigmented, just like every other part of the uvea is. But the choroid is special because it's going to be the vascular layer of the eye. Now you might be wondering why the choroid has vascularity and also pigmentation. Well, the eye has to have a blood supply, so the choroid is what's going to bring blood out to the eyeball. But at the same time, pigments help to absorb scattered rays of light. So when light does come into the eyeball, it is going to be tempted to bounce around it here, and that would significantly obscure our picture. But fortunately, since the choroid is pigmented, those extra rays of light will be absorbed by the choroid, and we can have a very nice clear picture, provided that everything else is working properly. Now, continuous with this is going to be the ciliary body and the iris, and I'll discuss those shortly. But before I jump into those, I'm going to introduce the retina, which is this yellowish layer that wraps around the inner 3/4 of the eyeball. The retina is what's going to contain the photoreceptors, which are the rods and cones. And the whole purpose of the retina is to get all of the light rays coming into the eye focused on one point and having that one point on the retina. Depending on where that focal point of light is contacting the retina, that's going to change and determine the picture that we will see. You can see the retina looks as if it's continuous here with the optic nerve. And it is in fact going to be connected because photoreceptors are just specialized types of bipolar neurons. Rods and cones are just bipolar neurons. So they're going to be projecting their axons backwards through the optic nerves, through the optic chiasma, and eventually back to the occipital lobe of the brain. So hopefully you can see these three different layers as they go from outer to inner. We also already discussed the cornea, but one we haven't really discussed much is the ciliary body. And the ciliary body can be defined as the enlargement of the choroid as it gets closer to this large blue thing, which is known as the lens. So the ciliary body can be found here. The ciliary body is going to contain a very special muscle known as the ciliary muscle, and that ciliary muscle is going to constrict to pull on these little fibers that are attaching here to the lens. These little fibers are known as the zonular fibers, and since they attach to the lens, whenever this ciliary muscle contracts within the ciliary body, that's going to pull on those zonular fibers and change the shape of the lens. The shape of the lens is going to change as we go from near vision to far vision. So this will in fact be continuous with the choroid and part of the uvea. Now the eyeball in fact is not a solid structure. There is in fact a space that can be found back here and also up here. Up here we'll have what's known as the anterior chamber, which makes sense because it's more anterior to this chamber, and back here we're going to have the posterior chamber, which is a bit larger than the anterior chamber is. The anterior chamber contains a specialized fluid known as aqueous humor, so that's going to be found in here. And the posterior chamber is going to contain a different fluid known as the vitreous humor, which is going to be found back here. The aqueous humor up here in the anterior chamber is going to nourish the lens and also help to maintain pressures within the eye. The vitreous humor, on the other hand, in the posterior chamber is more gel-like, and it's going to help in the transmission of light from the lens back to the retina, and also simply to help the sclera maintain the shape of the eyeball. The last few anatomical structures we have here for the eye are the lens, the iris, the pupil, the optic nerve, and the optic disc. Now I've mentioned the lens before. I mentioned that it's attached to these zonular fibers, and it changes shape based on the contraction of the ciliary muscle. But the lens's main role is to refract and focus light to a single point and to place that point right on the retina. The cornea will do a slight amount of refraction, but it is primarily the lens that's going to make sure that focal point is right on the retina. Now the other layer that we have here in the uvea is the iris. And the iris is in fact my favorite part of the eye. The iris gets its debut in many love songs, as this is the colored portion of the eye. But that is not why the iris is my favorite part. I enjoy the iris because the iris is responsible for regulating the amount of light that is allowed into the eye. So the iris is going to be responsible for constricting and also dilating, to decrease or increase the amount of light that is permitted to pass through the lens. In the event that we're trying to look far away, the iris is going to dilate so that we can allow more light into the eye so that we can see further. On the other hand, when there's a very, very bright stimulus, or perhaps we're looking at something close up, the iris is going to constrict to allow less light through the eye. Now, the iris is going to be doing the constricting and dilating, but really what the iris is doing is changing the shape of the pupil. And the pupil is the dark circle in the center of the eye whose shape is regulated by the iris. So the pupil, in fact, is simply just a hole. Light is going to come into the cornea, it's going to pass through the aqueous humor, and eventually pass through the pupil. The iris is not going to allow any light rays through it, so any light that comes into the eye must go through the pupil. Now, as light passes through the pupil, it's going to pass through the lens and get refracted onto the retina. And once our photoreceptors are activated in the retina, they're going to send signals back through the optic nerve, which is cranial nerve number 2, and that information is taken back to the occipital lobe of the brain, where a picture will be processed, giving us the ability to see. Now, the last structure we do have here I want to mention is the optic disc. Some of you may have heard of the blind spot and perhaps thought it was a myth, but in fact, I'm here to tell you that the optic disc is the blind spot, and we do have one. And there is an activity in your textbook, in the Special Senses chapter, that will give you the ability to find the blind spot. But essentially, because this is where all of the axons of our photoreceptors are leaving the eye, there's no photoreceptors on this space. So therefore, there can't be any rods and cones here to receive light, and therefore, we get the blind spot. So that will sum up the anatomical structures that we need to be aware of in the eyeball. I know that eye anatomy can be a little dense, so if you do have any questions about this or you're struggling with this topic, please make sure you bring that up to me in a Canvas message. Now, I was just talking about the blind spot and also the retina. I had mentioned that the retina contains photoreceptors, which are the rods and cones. So you'll see the rods and cones described here on this slide. Rods and cones are two different types of very specialized neurons, and they allow us to see two different types of light in two different situations. So rods are going to be the photoreceptors that give us the ability to see in dim light. So if you're out at night in the middle of the forest, you'll be primarily using your rods to see here. I don't know why you would be in the forest in the middle of the night, but in the event that you were, you'd be using your rods to see in that example. Cones, on the other hand, give us the ability to see colors. So a nice way to remember this, the C in cones stands for the C in color. And there are in fact three types of cones, specifically blue cones, green cones, and red cones. Now, what we'll actually come to see about these cones is deficiencies in one or more types of these cones will give different types of color blindness. And as we transition into our methods for vision testing, there is in fact an individual test for color blindness as well. So we'll get there in just a second. But the first vision test that we need to keep in mind is for visual acuity. This would be the vision test that you would have done throughout grade school using the Snellen eye chart. And for those of you that aren't familiar with the Snellen eye chart, it's the chart that looks like this. Generally, in order to assess vision with this chart, we'll tell the patient to cover up one eye and to read the letters in order in one of these lines. Now, what you may have noticed over here is that there's 20 over 100. We have 20 over 60, 20 over 30, 20 over 25, and 20 over 20. So, when assessing a patient's vision, we'll tell that patient to stand 20 feet away and again to cover up one eye. Individuals at 20 feet that can read this line will have 20/20 vision. If at 20 feet, these are the smallest letters that you can read, you'll have 20/30 vision. If it's this row, you're going to have 20/50 vision. So when we say 20/20, what that means is that you are able to see at 20 feet what somebody with normal vision can see at 20 feet. In the event that you have 20/100, you can see at 20 feet what somebody with perfect vision can see at 100 feet. So this is how we will utilize the Snellen eye chart, and once again, this is checking for visual acuity. The Snellen eye chart will test many different things, including the lens, the ciliary body, as well as the length of the eyeball. Now, the length of the eyeball is a very important determinant for how we're going to do when being assessed for visual acuity. So we're going to talk about that next. So this is a concept that some of you may be more familiar with, which is nearsightedness and farsightedness. You might in fact be nearsighted or farsighted, and what this means is that the eyeball is either longer or shorter than it's supposed to be. So, normally, how eyeballs are supposed to work is light rays are going to come in, they're going to get refracted by the cornea onto the lens, and then the lens is going to further refract those light rays to a single point right on the retina. So the focal point is on the retina in a normal eye. But with nearsightedness and farsightedness, that's not going to be the case. I myself have a very severe case of nearsightedness. So I have myopia. Myopia is our word for nearsightedness. And what this means is that the eyeball is a little longer than it's supposed to be. But even a little bit can make a big difference. So what's going to happen here with a myopic eye is the eyeball is a lot longer. Well, what this means is that the retina is going to be further back and out of place from where it's supposed to be. You can see that the focal point is actually exactly where it's supposed to be, but it's the retina that is not in the right space because the eye is elongated. So the focal point ends up in front of the retina, which you can see right here. Nearsightedness means that you can only see objects that are close up or near. Farsightedness is the opposite and means that you can only see objects that are far away. The technical term for this is hyperopia. So, hyperopia is the opposite of myopia, where the eyeball is going to be too short, and therefore the focal point will be behind the retina. Once again, the focal point is exactly where it's supposed to be. It is exaggerated in this picture just to give you an idea, but in fact, it is the retina that is out of place, not the focal point. So myopia and hyperopia are our first two pathological conditions that relate to the eyeball. You also may have heard of astigmatism before, and astigmatism is simply referring to an irregularity in the cornea. Maybe the cornea has a bump or a groove in it that ultimately produces many different focal points. If the cornea is not the right shape, it can produce many different focal points, and this is also going to cause difficulties with vision. You can see a normal emmetropic eye here, where the light comes in and focuses on the retina with one focal point, but here with astigmatism, we get more than one focal point. Now there is another test that we can use to assess for astigmatism, and so we will use this Astigmatism Chart. It's not a very fancy name, we simply call this the Astigmatism Chart. Now what's going to happen when we're assessing for astigmatism, we're going to tell our patient to cover up one eye and to look at all of the bars presented in front of us. And provided that we don't have astigmatism, all of the bars should be about the same width. In the event that some of the bars are thicker than others, that is a possible indicator of astigmatism. Now, just a disclaimer, I told you guys at the beginning of the class that I am not a doctor, and I am not out here handing diagnoses for all these different conditions. I just want you to understand that I am well aware that some of these bars are in fact a little thicker than others in general. It is the concept that I want you to get down, and if you're interested in getting a diagnosis, go to the eye doctor. But essentially, that's how this is supposed to work. If we cover up one eye and the width of the bars increases or decreases, that is a potential indicator for astigmatism. So I have already mentioned color blindness, and I mentioned that there are a few different types. Color blindness occurs when there's an absence or perhaps a deficiency in one or more types of the cone. Remember, there's red, blue, and green cones, and if one of them has been knocked out in some way or is not functional in some way, that's going to lead to color blindness. We do have ways that we can assess for color blindness, and what we are seeing here are Ishiharas color circles. Now, I always find it important to describe these in case any of you are in fact colorblind. What we have here are some circles that are in different colors, but there's a number. You can see this is 13. We have 13 right here, we have 16 over here, we have 9 right here, and so forth. So we have different numbers that are found here within the circles, and depending on the type of color blindness, that's going to be indicated by the particular colors in a particular circle. Alrighty, so next up is the near point of accommodation vision test. The near point of accommodation, or the near point of vision, is how close you can see something with your eye before it becomes blurry. This is going to assess how elastic the lens is. Now, as we get older, the elasticity of the lens does tend to decrease. We call this phenomenon presbyopia. Of course, it's going to be closer in infants, but normally about 10 centimeters away from the eye is where we'll find the near point of vision. So in order to facilitate the near point of accommodation or near point of vision test, you'll take a sharp object such as a pin or the head of a ballpoint pen, and you'll look at it from a distance and slowly bring it closer to your eye. As it gets closer, it's eventually going to get blurry. And at the point that it gets blurry, that's going to be your near point of vision. Normally, as I mentioned, this is about 10 centimeters. But as we get older, this will get worse, and in infants, it will actually even be better than 10 centimeters. So our first visual reflex that we're going to be able to test for is the Accommodation Pupillary reflex, which is facilitated by taking the patient, putting them in a dark room, and taking a flashlight and putting it right in their eye. Normally, with distance vision, the pupils are going to dilate to allow for more light to come into the eye. The pupils are going to constrict with near vision when we're trying to look closer. So in order to check the Accommodation Pupillary reflex, you'll put your patient in that dark room and tell them to look off into the distance. You'll then take that flashlight and point it in their eye, and what you should see if the reflex is functional is the constriction of the pupil. As more light is shown into the eye, the pupils are going to constrict to make sure that no light is going around the lens. Keep in mind that the lens is going to be changing its shape, and it bulges when we're looking up closer. When the lens bulges, it's going to be a little bit smaller. So as we take the flashlight and put it in the patient's eye, that's an overload of light, and therefore the pupils are going to constrict to prevent too much light from entering into the eye. So finally, to finish up this special sense, we have one more examination that I need to introduce you to, and that is the ophthalmoscopic exam. The ophthalmoscope is this device shown right here. And essentially, the eye doctor is going to use this to look through the pupil and make an examination of the retina. There are lots of structures in the back of the eye that are going to indicate the health of the eyeball in general. Of course, the retina is what we're looking at, but we'll also be able to see the optic disc and the blood vessels behind the eye as well. The optic disc is supposed to have a very clear demarcated line around it, and if that line starts becoming fuzzy, that can indicate some issues with the optic disc and the optic nerve. On the other hand, the blood vessels that are found within the choroid, if those are enlarged, that can be an early indicator of hypertension. So there's lots of different things that we check for using the ophthalmoscope. I've put the picture here just so that you guys can get an idea of what the ophthalmoscope is and how it works. Now, as you continue to go through the special senses, we're still going to go over olfaction, gustation, audition, and equilibrium. One suggestion that I would like to offer you all is to make sure that you're going through the lab manual sheets and looking into the activities and also into the additional information that's posted in those lab manual sheets. This is one of the weeks in particular that the lab manual is going to be especially useful. So make sure that you have yourself a copy and that you're filling these sheets out as you go through the special senses unit. That will conclude our first part of the special senses presentation, and I'll see you all in our next lab recording where we'll start discussing the ear.